So good morning, everybody. Welcome to Medical Grand Rounds. Before we begin, I would like to respectfully acknowledge that we are located on Treaty 6 territory, traditional gathering place for diverse Indigenous peoples, including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Anishinaabe, Inuit, and many others whose histories, languages, and cultures continue to enrich our vibrant community. I'm delighted to introduce one of our new additions over the past few years. Dr. Vanessa Myers-Stevenson is an infectious diseases physician and clinician scientist with an interest in chronic viral hepatitis and the study of viral host pathogenesis.
You're good. I can.
Okay. For this and other viruses. Her research lab uses a combination of computational, biophysical, and molecular virology methods to explore various viral DNA, RNA, and host protein interactions to gain further understanding in viral pathogenesis and evaluate unique approaches to developing therapeutics. That's a mouthful. So it's a very, well, it's a very important work. So without further ado, I'm going to ask Dr. Myers-Stevenson to come talk about an update on hepatitis B virus, where we are and where we could be. Thanks, Vanessa.
Thank you for that introduction. And I'll turn on this speaker thing here in a second. Make sure I've got the sound in the room. Oh, there we go. Oh, that is really loud. I'm gonna move that lower down. Yeah, we'll try that. All right, at least if I move, then at least the mic will come with me. Okay, so thank you again for the invitation to tell you a bit about this stuff. I've got a few different sort of plans in mind for objectives, and I'll go through some of those here as well. I'm gonna just turn down the volume. Great, thank you. And so again, I also have a land acknowledgement slide here as well. We are working on, we do our research on the Treaty 6 territory. Okay, so to show you those objectives, so I'm going to review some of the impacts of Hep B and the role of screening and vaccination. And then I'm going to talk about some of the challenges around hepatitis B cure, and then dive into this sort of stuff we're doing in our lab. Very, very quick elevator pitches on that front, because I could certainly talk many hours on that topic. Quick disclosure, this is largely for the stuff I'll talk about with my research, but I do have a patent on a class of drugs for HBV binders with under development, Quadromix Biotech. And I've also worked in labs where they have connections with different studies and whatnot, but none of those have influenced that work, and nor is the trial that I'm currently on drug company funded. So hepatitis B affects nearly 300 million people worldwide, and if we look at the Canadians, that would correspond to, that's about 300,000 Canadians. So it's a fairly significant number. We actually have limited Canadian data, and limited data doesn't mean a limited problem, or lack thereof doesn't mean a lack of a problem. And one of the studies or surveys a few years back, or I guess it's up a decade ago, showed that, you know, upwards of probably 50% are even unaware of their status, and as far as that goes. In terms of how hepatitis B ranges and change or varies across Canada, there's lots of variation, and this study here in 2013 showed some of the genotypes and how they vary across Canada. And if we needed one more reason to say that Calgary and Edmonton are very different, here we go. Even our hepatitis B strains are different. But this likely has to do with where people are from and where the people have immigrated from. And so the various genotypes of hepatitis B, there are currently 10 altogether, but they tend to have various epidemiological locations. So hepatitis B is a silent killer, and this is largely because of the cirrhosis and HCC that it causes without essentially any symptoms leading up to it. And so to kind of go through some of this in detail here, we have a healthy, infected but otherwise healthy liver that, if over time it develops inflammation, that inflammation can eventually lead to scarring or fibrosis, that can lead to cirrhosis, that ultimately can lead to HCC. That rate of cirrhosis to HCC is over 5 years. So those who have this can develop 15 to 25% over 5 years will develop HCC. Of again, the cirrhotic patients, 15 to 20% will go on to decompensate, having these complications noted here. And then over a 5-year period, again, if fibrosis and scarring is severe enough, again, can progress to these stages. Now, treatment and intervening can actually reverse some of these effects, and this is partly some of the reasons around this. So unlike hepatitis C, where you go through those stages of fibrosis, cirrhosis, and then cancer, hepatitis B also has a unique way or a unique route of creating cancers, and this is because it has the ability to integrate into DNA. It is no longer infectious once it's integrated into DNA, but it can still produce some proteins. And if it integrates into an area that has an oncogene, it can actually create a little island of a malignancy. And so that's one of the things that is unique about this virus. So that occurs at a rate of about 1 to 3% over 5 years. So it's not insignificant in and of itself, but this is sort of some of these things that makes hepatitis B so silent and so hard and challenging to go after, because you have young, healthy people that are developing these cancers or having these things, sometimes without the signs and the prelude to it that can that give you the hint of, oh, I need to follow this person more closely. Some of the clinical factors that link a person's risk to developing HCC are both clinical, so age, male sex, alcohol use, and frequency of ALT flares. But there are other viral factors as well, so genotypes, so genotype C being the most common one to progress to cancer, different viral factors and co-infections. And again, that annual death rate with HCC is 20 to 50% once people get into this decompensated state. This is the rates, a snapshot of the rates across the world in terms of HCC. And just to kind of highlight some of the significance of this on a global scale, in low income settings, if you're diagnosed with liver cancer, you will have a prognosis of just within a few months for that. If you're in a high income country, so here in Canada, several months to a few years. So certainly nothing to ignore from that perspective. But it has huge implications in terms of how we kind of go after this. The World Health Organization has a strategy for elimination of viral hepatitis. This includes B and C. Their goals were probably a little bolder than people have probably realized that is doable, but you have to aim big. But their elimination goals were to eliminate viral hepatitis as a public health threat by 2030. And that's defined as a reduction of 90% of new chronic infections and 65% reduction in mortality compared to the 2015 baseline. So a table for this here is just kind of showing some of this to show for hepatitis B, our baseline coverage, 82% for 3-dose vaccination, and their target was to get it up to 90 by 2020, 2030, up to, maintain those levels. I'm not going to go through the table. But essentially, just to summarize there, for hepatitis C, so that's not B, hepatitis C, there are only 11 countries that are on track for meeting that 2030 goal. Canada is one of them, so that's fantastic. So that's for hepatitis C. For hepatitis B, worldwide, there is no country that is on track to reach that target, which is clearly an issue. But I'm going to give you a bit of information about what we can do to kind of help things along and kind of bring a bit more voice to hepatitis B. And this is the extension of that table there. Okay, so hepatitis B, like I said, it's a silent killer. It's a silent killer because of the virologic sense and the clinical sense. It doesn't actually cause a lot of symptoms, so you don't know about it often until it's too late. It's silent in the social sense. It is an illness that largely affects those in lower socioeconomic status countries and populations, and in Canada, it's largely the immigrant population. And so they already have a quieter voice in terms of our healthcare and some of those strategies. And so as well, the stigma that gets associated with bloodborne infections, people don't want to talk about it, people don't want to admit to it, people don't want to share it with their family, whether culturally or for other reasons from that front. So it is challenging. And then there's the lack of awareness. So myths, misconceptions, and maybe complacencies. But so we have a vaccine and it's effective and therefore it's not an issue, especially not in Canada, right? So that's the you know, the thought around this. So yes, we do have a vaccine and yes, it is very effective. But I'm going to show you some data on that now as well. So diving back into some of the risk around exposure for chronic hepatitis B, as a neonate, so as a baby, you have a greater than 90% chance of that exposure turning into a chronic infection. If you're an adult, let's say you've never been vaccinated, everyone in this room is probably vaccinated anyway because you've come through the healthcare system and your training and whatnot. But if you're an adult and you get exposed and you haven't been immune, you're not immune, you only have about a 5% chance of that exposure turning into a chronic infection. So it's a complete flip-flop of the numbers in terms of your risk and the risk of the chronic infection. So this is why we should have a universal birth dose vaccination. This is actually a WHO recommendation on that front. What that would do is actually would greatly reduce the potential for neonates and babies to get chronic hepatitis B. So with a woman who is e antigen positive and high viral loads, she has a baseline risk of 90% chance of passing that on to her baby. But if we give the birth dose vaccine, that already drops down to 21%. If a woman is e antigen negative, that already has much lower numbers there. Just showing that 30% down to 2.6. Now, if we also add in hepatitis B immunoglobulin, we can decrease that even further. Just telling some of my patients some of these numbers, and so who are pregnant, just the shame that gets associated with having the virus and potentially passing it on to your child is just undescribable. You kind of hear some of the stories around some of this, but being able to say, no, this is what we're going to do. We're going to be able to help you prevent this transmission to your child, and like just the relief on some of that is just phenomenal. And just being able to do that for people. But we have to do it, and we can't do it unless if we don't know about people's status, right? So this is the other thing. So the next question is, so how are we doing here in Alberta? Yes. So before 2018, we would be doing hepatitis B vaccinations in junior highs. So students would be getting in grades 6 or 7. But since 2018, we're now kind of getting a little bit closer to down that range. We're now doing it at the 2-month visit, so after birth. And so we're like, okay, so we still have ways to go. This is the Canadian Hep B vaccine schedule just for the different provinces. So how are other provinces doing? So there's actually 1 province and 2 territories that are actually meeting WHO guidelines. So New Brunswick, Nunavut, and Northwest Territories. Everyone else has their own variation on when they vaccinate children and all that. Why is that an issue? Well, clearly because of the neonatal potential for exposure. And then I'm going to also bring out this other study that was done in Ontario. So this is a study that the title is from a subset from myths. So this is why that's listed as an effective HBV vaccination program. The study looked at children in Ontario over a 10-year window with hepatitis B and whether they were Canadian-born or foreign-born. So if they were foreign-born and they emigrated to Canada, well, that's one thing. We wouldn't have had a chance to intervene there. But if someone was born in Canada, if we had birth dose vaccination as a standard, look how many children we could have prevented from from being exposed to hepatitis B. 139. That's almost shocking. 139 children born on Canadian soil that we could have prevented. That's just in Ontario. And so this is something that, if we had a universal across the country plan, and maybe it wouldn't be 100% of protection, but even that, the numbers with that we showed there could have a huge impact. FYI, for those residents in the room, this is a research opportunity. We can actually replicate this data for Alberta. It's actually fairly quick turnaround. Just got to get the ethics through. But then just an FYI, email me. So for everyone else in the room and online, okay, you're saying, okay, yeah, that's great. I'm not an obstetrician and I treat adults. So what are you talking about? How is this relevant to me? Well, this is where I get into the whole, you can also do this for your other patients. So this is consider screening, once a lifetime screening for all your patients. So whenever you think of doing this for someone with HIV, just throw in the triple screen for hepatitis B. Super simple, then it's done. And then like what this is actually also endorsed endorsed by the Canadian Hepatitis B Network. So it makes for a fairly straightforward and a simple thing that you can do, just even raising awareness. So just to kind of throw in an anecdote, of course, from this, but you know, I had a grandfather come in with their daughter in my clinic, and the daughter was there for translations. And as we were talking about this, she came to realize, you mean Grandpa has hepatitis B and could have passed it on to me and my kids for the past 10 years, and we didn't even know about it? And it's like, well, were you given all of the screening for public health when you came here? I was like, yeah, but I didn't think anything of it. And so this is the thing. So even just having one member of the family aware of their status can then prompt others to learn about it and to become educated about it, and then to get screened for it. And so every individual, even if they are much older, it has the potential to be connected to many other people, right? I mean, that's the thing, where no man is an island. So then I'm going to move on to my second objective now. So to discuss some of the challenges around HBV cure. Okay, so we have treatment for hepatitis B. That's great. Treatment is not cure, however. Treatment is suppression. So our current antivirals are interferons and nucleoside analogs. Oh, if I shake this enough, I can get the mouse to show up. Okay, so nucleotide and nucleoside analogs act at a step downstream here in the viral process. And it is very good at suppressing viral loads. That's great. But unfortunately, the problem is the chronic form of the virus, known as covalently closed circular DNA. So this little structure here inside the nucleus is actually hidden away and protected from all of the other signs in the immune system. And so we have the ability to reduce this, but this cccDNA still keeps pumping out little bits of virus into the cell and into the bloodstream. So like I said, we have multiple nucleotide analogs, including one developed here by Dr. Lauren Terrell, lamivudine. This was the first hepatitis B drug, also now used for HIV. But we don't tend to use this one anymore because of the resistance that is developed. But just the same, it started the whole process of, can we get things going, and can we start, creating more drugs for hepatitis B? So as I alluded to here, that the cccDNA, I'm going to show this electron micrograph that I had at the front. I really like this one because it really shows kind of the whole scheme of what we're actually dealing with here too. There are 3 full, complete viral particles here, and that's the infectious virus. But then there's all of these other little bits. These are all of the empty particles, subviral. This is rich in surface antigen. So the virus actually spits out lots of this surface antigen as a decoy for the immune system. So it's able to kind of keep the immune system at bay, so that these little viral particles can get into where they need to go to start replicating. And so that has the implications around that. So again, while we are treating with medications, yeah, we can reduce that, but we still have lots of surface antigen, lots of these other things. So the immune system is still being challenged by the hepatitis B. So one of the challenges then being with hepatitis B cure is, well, first off, how are we going to define it? So defining cure, there's essentially 2 definitions. There's functional cure. So there's the loss of HB surface antigen, so loss of all of those little particles, which means essentially that cccDNA is no longer pumping out or spitting out all of these little viral particles that are causing all the trouble. So that's one definition, and that actually is a very strong definition from in terms of reaching this functional cure, because once you reach that stage, your viral markers all are normalized, and your risk of complications of hepatitis B are actually so low that you no longer need to have the Q6 monthly screening for ultrasounds and the blood work for monitoring. So when patients get to that stage, it's a really good point, because then then it's almost like you take control of your life back again. You can just ignore it for a long time. Getting there is not straightforward either. So spontaneous loss. So some people will just clear on their own. So they'll develop antibodies to the surface antigen. And that happens at a rate of about 0.5 to 0.8 per year. And interestingly enough, treatment with nucleotide analogs and interferons and all that, despite all the things that we do, it still doesn't change those numbers. So that really is curious first off, why that, if we lower the viral loads, why doesn't it do something further? Why doesn't it improve the outcomes on that front? And there's still plenty of room for research and things to be looked into and from that front. So what's the other definition for hepatitis B cure? Well, the other one is virologic cure. That's where that cccDNA inside that nucleus, that's destroyed or removed. So that's the two different definitions from there. So what I'm going to refer to going forward is actually virologic cure, because that's the one where I think we can make some gains and get in using knowledge from multiple fields to kind of really put a little bit of emphasis on. And just to purposely show this busy slide, the hepatitis B cure pipeline, partly to also kind of raise awareness, and also just to kind of say, give additional hope to, patients who you have that also have hepatitis B, just saying, there's a lot of people working on this. There's a lot of people in different areas, and there's a lot of discussions around what can be done and where we might be able to go with this. So just to kind of give a bit of hope on that front, that it's not necessarily out of the realm of possibilities. A few of those I find really interesting here that, well, Merculodex is already one that's being used for hepatitis delta. It's an entry inhibitor, and it's in phase 3 trials in Europe, and we have some people here in Canada on it. And then we have some of these other ones, but the one I'm also going to focus on, of course, is G quadruplexes. So the direct cccDNA targets, and this is some of the stuff that we do in our lab. Leading me to the third objective. So I'm going to take a few minutes to talk about some of the things that we do in our lab around the hepatitis B therapeutics development. So I'm going to briefly talk about two different projects that we have in our lab. One is the quadruplex study, and one is the Hep Delta HDV mimic study. And so I'll dive right into the first one here. This one has a bit more to it for now. So back to that little picture of the cell with our cccDNA in the center. If we want to go after this here, the first thing we have to do is, well, find it. We have to be able to find it and bind to it. So that being the first sort of challenge of this, okay, well, how are we going to do that if we're going to go after this thing? Well, let's take a closer look at it. So the virus itself is actually quite tiny, 3.2 kilobases in size. For comparison, coronavirus is 10 times that, so 32 kilobases. So this is a very, very tiny virus. It actually, all of its proteins are in overlapping reading frames, and it only produces 7 of them. And the 7 are actually really only 4, because 3 of them are the same isomers. So it's tiny. The cccDNA is actually supercoiled and wrapped around histones. So for all intents and purposes, it looks just like our own DNA, and it's hidden away in there and in the nucleus. And so going after this has multiple challenges. But if something is going to replicate, what's the first thing it has to do? It has to open up, it has to loosen up from those histones, and it has to expose its promoter regions. And so this is kind of the target and the justification for how we're going to go after this in terms of the virus. So take an even closer look at this. One of the promoter regions here, this is a pre-core promoter region, one of the more critical ones to the virus. So I did a review of all of the available sequences available from our hepatitis B database. Now, this is clinically derived samples, and therefore, by extension, replication competent virus samples. And what that implies and what that shows is that amongst all the genotypes, amongst all the sequences that are available in that pre-core promoter region, there was a section that was so highly conserved across all genotypes with a high predominance of guanosines from here that for all of those guanosines in themselves, over 99% conservation across all of these sequences. So huge implications for what its importance is to the virus. And just kind of highlighting that in another way, this region is so highly conserved that even in a mummy that was exhumed from Italy from the 16th century and had hepatitis B diagnosed, they were able to actually also isolate some of the virus and sequence it. 100% conservation. So this region, despite the fact that hepatitis B has been around for centuries, has really not been able to mutate out that region without making itself replication incompetent, right? So this region is so important to the virus that it's had to keep it for centuries, quite literally. why is G-rich regions important from this perspective? Well, G-rich regions can actually form what's called a quadruplex. Okay, so diving into that term, a quadruplex is when you have planes of 4 guanosines that stack upon one another in different conformations to create these 3-dimensional structures. What that means is that this is a targetable 3-dimensional structure, so therefore druggable 3-dimensional structure. The sequence that is typically required for a quadruplex to form is shown here. So we have groups of 2 to 3 guanosines or more separated by nucleotides anywhere from 1 to 7. Sounds like it could be pretty random. And there are actually quite a few potential G quadruplex sequences found throughout the human genome. But it's not just randomly found. They actually have a high predominance in promoter regions, in telomeres, in regions of IgG switch, in high DNA functionality in our genome. So there's a lot being learned and a lot being explored in quadruplexes in a lot of realms. So in the cancer world and immunology. So this is just one more world where we're like, actually, you know what? These probably are also very much a functional part of viruses. And so when we're looking at this here, diving into that with that in mind, we dove into the older literature and we found some work that was previously done by Lianu in 2001. Where they showed that if you make a few mutations in this region, you end up losing the core transcript from there. So we thought, okay, well, you know, maybe that makes quadruplex, and so we made the same mutations and kind of looked into some of that. So if in that promoter region, the transcription factor SP1 binds to that area to promote the transcription. So making those mutations, we found that, okay, yeah, actually, the ones that they had in their paper also the wild type did form a quadruplex, while the single mutation actually started to unfold it. And so that would in and of itself then made us or led us to the hypothesis that actually, yeah, so maybe that is a quadruplex that's in there, and that's actually what recruits that SP1 to help the transcription. So looking into that a bit further with more mutations and more insight into all of this, we did some of this work, and I'll just kind of show all of these kind of in a snapshot. This is some from my postdoctoral work. But looking into different mutations and analyzing to show that, yeah, they are actually forming different things. This is the circular chromosome, so it is unfolded. And then binding studies, we can show that, yep, it binds to another quadruplex binder, and we can actually pull it down from a liver extracted CCC DNA from that front. And then when we transfect it into cells with the wild type and mutant, we see that there's no difference in the surface antigen, as that's not affected by that promoter. But when we look at wild type versus the mutant here, so pink being the wild type, my mouse isn't showing. Okay, pink being the wild type shows that, you know, you get normal levels, but then the double mutant greatly reduced. And so this is what's led to the development of this protein-based approach, so a nanobody approach for a quadruplex binder that is still being worked up and worked through by the University of Lethbridge and University of Calgary with the teams there. And has led to the patent that we have on this class of binders. And this table here shows like the panel of 11 different ones that we had developed and shown that actually, you know, for some of these, there's actually a great degree of selectivity. So that is important because being able to be selective with your targets is really great when you have a lot of different quadruplexes that are found in the human genome. So that was from my postdoctoral work, but when I came to the University of Alberta, I broke off to kind of do some of the small molecule work. Why small molecules? So small molecules are more druggable. You can do different things with it in terms of rational design, being able to kind of control different things and factors, and do modeling to be able to say, okay, really design out the toxicity, design in more selectivity. And so has that ability to do some of this. We've all kind of agreed on terms of where we go with all of this, but they're like, okay, yeah, you take the small molecule. We don't want to deal with that. And so like, perfect. I'll turn it into something bigger. And so in our lab, we actually have a 3-pronged approach to this when we're doing our work on our small molecule strategy. So computational work, biophysical work, and the virology. This is all integrated into one another, and the data from one feeds into the other to make sure that we're keeping it relevant and that we're focusing our energies on the right targets as we're doing this. And to really just show the quick snapshot, I'm just going to check the time. Yep, okay, we're good. Quick snapshot of kind of where we are with these things. just to kind of show, we're currently creating a model. This is using, one of the things is with AlphaFold, because there is no structure right now of SP1. So this is one of the things here, using with our quadruplex. And I have 2 people that are working on that to try to see if we can get that going. We have our SP1 production, so that's the transcription factor that we're producing in large enough quantities to be able to do biophysical studies. And so we have the people here that are helping with all of that. And we also have a team that are working on the functional studies, so the localization piece and drug testing with compounds that are actually been developed as binders for the cancer world. And so we're using that as our starting point, and then kind of going from there. Maybe we can modify it, maybe we can make the cancer drug more selective, and kind of, but it gives us a starting point to even just be able to model and to work through some challenges. And because I want to showcase some of my other undergrad work that they've been doing this past summer as well. Oh, I put it all at the same time. So I had one of my students, Daniel Wang, made the reporter plasmid. Yeah, there we go. Made the reporter plasmid. So the luciferase is under the control of our pre-core promoter. So this is the area that contains the quadruplex. And then my other students were working on making different mutations and testing those in the luciferase model. And we can show that, yep, if we make some certain mutations in the loops, we end up with variations in the amount of output for our luciferase. And then similarly, we were asking the question, well, okay, so that's our current quadruplex. What if we swap it out with any quadruplex? What if we take one from the telomere and pop it in? What if we take one from the BCL2 and pop it in? Does it make a difference? And so there's actually some that are known to be SP1 binders and some that don't bind SP1. And we've been working on kind of looking at some of those. And so actually, yep, it is also selective in terms of how it binds to certain things. So there's more to it and lots to come from that front. But ultimately, this is all feeding into our computational model to make sure that it's accurate and it's kind of working, but ultimately we do want to get a higher resolution structure. And this leads into some of the great resources that we have currently here at the University of Alberta, because with the recent government funding for pandemic preparedness and whatnot, we are going to be acquiring a cryo-EM this year. The physical machines are in crates and being assembled, and all of these things are happening right now. So this also links with a Canada Excellence Research Chair. So we've recruited a Belgium expert in cryo-EM to also help with the program. And this is now part of what we call SPARC, so Striving for Pandemic Preparedness Alberta Research Consortium. It's essentially kind of the way that we had taken the funds to be able to turn this into a sort of a strategy for pandemic preparedness going forward. So if I'm going to summarize with that quadruplex study, just with this piece here. So hepatitis B, as I've already well alluded to, is a chronic because of that cccDNA. And if we're going to go after cure, we need something that's going to go after that structure. And our lab is currently looking at a way to selectively bind and anchor to that quadruplex. So what implications will that have if you can actually bind to it? Well, first off, if it prevents transcription, then great, we've got another potential drug to be able to help with a further step upstream from what we're currently doing. So that in and of itself is useful. But if we can attach a payload to that specific binder, we can actually target that with different strategies and tag team it with other researchers' work to be able to make something more specific. Specificity is one of the challenges that a lot of people have had with some of the different strategies, like with CRISPR-Cas9 methods and whatnot. So attaching a payload has a potential that is perhaps not necessarily considered or thought of in terms of the strategy from all of this. So stay tuned and keep for updates on this front. But like I said, ultimately, if we ever want to actually clear the cccDNA, we first need to be able to bind to it. And so then that brings me to the second project I wanted to just tell you about, another kind of a fun one derived from, just out of the box thinking on some of these fronts, hepatitis D mimic. So a lot of you know that you can't have hepatitis D unless you have hepatitis B as well. Hepatitis D relies on the surface proteins of hepatitis B in order to complete its life cycle or replication cycle. Let's not call it living. It's a viroid. It's a deficient virus. Of the people living with hepatitis B, there are about 5% of those that also have hepatitis D, so hepatitis delta. That equates to about 12 million people worldwide. So again, not an insignificant number. Now, interestingly, with co-infection, when you have both of these viruses in there, what often tends to happen is that the levels of hepatitis B are actually lower. So you get rip-roaring ALT flares and lots of inflammation, but actually the hepatitis B virus is lower. And it's actually by a method that's independent of all of the interferon response. So essentially, we have this viral competition happening inside the cell. Hepatitis D is winning out a lot of the time. And so it's suppressing hepatitis B's replication to be able to steal all of its surface proteins. And researchers in France, this is Dr. Julie Luciferoy from the INSERM, presented this at one of the international Hep B meetings, and describing, okay, actually it is independent of interferons, and we think it binds to the pregenomic RNA, and so this area where the protein requires the DNA polymerase to bind to enable encapsulation, so the epsilon region. So if you can't bind to your epsilon, you can't encapsulate, and you can't you can't use your proteins to be able to exit the cell. And so they showed that one of the proteins that hepatitis D produces, binds to this area, or supposedly binds to this area, but they weren't able to take it much further. And so we're like, okay, well, we can do the structural piece, we can do that structural workup. And so we're working together to try to get the pieces of this coming together to be able to really tease out what's actually going on here with this. And so the first step, of course, would be to take this, make the protein. So we cloned it and produced it. And I've got two students on that. One summer, well, they were both summer students at the time, but now Justine is now one of our graduate students. We're making that protein here. It is confirmed by mass spec. The next thing was to do to figure out, okay, well, we need to make the epsilon, or this region here. And then the next question led to, okay, well, which epsilon should we make? Like, there's hundreds of thousands of different potentials that are from that are out there from these different strains of virus. And so one of the first things we're like, okay, well, first off, does hepatitis D have a preference for the type of hepatitis B that it goes for? Is there one genotype that pairs with the other more often? And so as we're doing this review, we decided to turn it into a publication to look at, okay, well, actually, so the most common combination is hepatitis D1 and B genotype D. And so while there could be some component of viral preference there, it probably more likely is related to the epidemiology rather than the actual the baseline viral components of it. But just the same, we wanted to make sure that if that was the case, then we went to take that same database of so in this scenario, we've got with 13,000 different sequences, and we actually looked for, okay, well, how much variation is there? And let's get a consensus sequence to get our region actually highly conserved again across the board, which was actually reassuring because we didn't actually have to lean on well, we'll pick this because it's the most common. Well, actually, this will be pan-genotypic. It's essentially common to all of the viruses. And so now we're on to making some of that there, and we're trying to do the binding and structural work. Right now, we're kind of in various phases. We've tried different things with infecting the cells directly, and have got my other students here doing some of that, infecting the cells. These ones haven't been successful, but potentially because of other confounders in the actual cells themselves. But we're trying various other things here. We're getting there with some of our in vitro transcription to be able to actually produce it to show that we can bind it, and then we'll work backwards again into the more complex systems. And ultimately, what we're hoping to do is to be able to create an HDV mimic. So effectively, we could create a new therapy for hepatitis B by pretending we're hepatitis D, which has its neat implications from that front. So in summary, and I think I'm well under time. Yeah, great. Hope we'll have lots of time for questions. So in summary, where are we and where could we be? So just to kind of go through those different objectives that I had here, we talked about some of the impacts of hepatitis B, the role of screening, which you can certainly do, and with vaccination. And so just bringing up that, yes, birth dose vaccination is really important. We're still advocating, partly as the Canadian Hepatitis B Network is still advocating with the government. Okay, we should put this across the board, and involving NACI and trying to say, what, let's just make this happen for everyone. Let's not make it let's take the thought process and the individual risk stratification off the table. Let's just get everybody vaccinated. And then the part that you can also take part in is screening people for hepatitis B. So the triple screen, right? The three things to order. We also talked about some of the challenges of hepatitis B in terms of cure, it being that chronic form in the nucleus, and the fact that it has all of these decoy surface antigen things that it keeps spitting out. That's why your body your immune system is getting overwhelmed by all of these decoys, and it's not finding the virus, and it has all these extra challenges. So there's a lot of components to this that, you know, even with great suppressive therapy, it's still not enough to get rid of it. And then we talked about some of the different things that we do in our lab, those 2 projects that I'd highlighted. And of course, none of this would be possible without my great team that I have in the lab. We're a big group, but all motivated and excited to kind of make headway on this and some of our other projects. Obviously, lots of people here at the University of Alberta that have supported me trying to getting this up and running, and I've got several people bolded here in terms of their contributions to really just help me get off the ground and get things established here at the university, and in terms of all of the nuances and challenges around starting a new lab at a new place and then during a pandemic and all of these other, you know, complications and things around that. So really grateful to all my mentors, and thank you everybody for joining in, listening to my talk. Hopefully you've picked up some tidbits that you can use in your daily life and excitement around hepatitis B.
Thank you, Vanessa. That was fantastic. A nice little update, and you know, in terms of where things are at with hepatitis B, but I also think it's a nice sort of presentation of how, you know, a career evolves with various parts that you picked up when you were doing your PhD work and so on, and the ongoing applicability and so, you know, continuing the work to ultimately the cure, right? Which is what you're hoping for. Yeah. And I wonder, just a couple of questions, one around the translational piece where you talked about, you know, sort of screening. So as a general internist, I mean, we have this population that we automatically screen, right?
Yep.
You know what I'm talking about?
Mm-hmm.
But then really it kind of stops there. So what would be your sort of general recommendations in terms of screening, you know, within a cost-effective, reasonable manner, and has it been looked at in terms of general screening and cost effectiveness?
Mm-hmm.
And then the second question I have is more around the applicability of what you're looking at with that CCC integration.
Mm-hmm.
Has that been looked at with other infectious or other disease states, and whether or not there would be applicability for treatment more broadly than hepatitis B, I wonder?
Okay, great questions. Okay, look, I'll talk about the screening piece, and then we'll dive into the basic science because I might take longer on this. Yeah. So with the screening, like I said, it is supported by the Canadian Hepatitis B Network in terms of the cost effectiveness. I'm not sure there's been a study that's really laid out the numbers. And yes, there could be a surge of then kind of testing requested by the lab from that front. The surface antigens and antibodies are actually, as far as some of those go, they're actually they're cheaper tests versus a DNA test. I mean, if we cross-check, okay, if they've had a previous positive and previous immunity, then no need to do it. I mean, at least now with our records, they're a little more robust, but I mean, I get it. People move around and we won't necessarily have all of the information. Yeah, the population around. So, okay, so now you've got, you know, your patients coming from the nursing home. Are we going to be screening those individuals? And I know it kind of comes down to some of those challenges from that front, especially as we age, become a bit more you have immunosenescence and the complications around that. Knowing if someone had a prior exposure could actually have utility if they were to develop complications around, you know, ALT flares. I get it. It's probably, you know, in terms of, you know, being able to intervene and prevent certain things in you know, older population may not necessarily be as, you know, as helpful from that way. And for the same reasons why we don't start statins on, you know, 90-year-olds. It's, you know, the you know, cost benefit that way. But then I take that step back again and make that argument that, yeah, you know what, that grandfather that came into my clinic that has children here, we're all connected, right? And so they're not going to show up to hospital because they're perfectly healthy.
Yeah.
But if grandpa gets tested and he finds out he has it and he's been exposing his family for the past decade here, I have a harder time arguing against it from that front. Yes, I agree. Anyone, of course, who's received blood products in Canada before the, you know, the screening programs that really went in, we have all of those sorts of things in place from there. So there's other still justifications around it, but yeah. So yeah,
and the question is really around generalized, right?
Yeah.
And then women of childbearing age and things like that. I think there's a lot of unknowns still there.
Yeah, right? Yeah, for sure. But it is part of our prenatal screening. But again, a woman has to get follow up and get prenatal care in order to get those sort of screening. And a lot of the population who are immigrants don't often show up because they don't understand what the system is and how much they'll have to pay to get the screening. And yeah, so a lot of misconceptions and maybe misinformation there, but also yeah, it's opportunities for making a difference in some of those fronts. Yeah, okay, so then the question around integration. So hepatitis B integrates, HIV obviously integrates, EBV can also integrate, but whether or not it's infectious in that form at all is another question from there. So when hepatitis B integrates, it actually, because it's a circular genome, it actually gets cut where in the pre-core promoter. So the pre-core promoter is no longer able to translate the rest of the virus. So therefore it is not infectious in its integrated form. However, all of the promoters are still intact for the production of surface protein. So technically, even if you were to fully clear the cccDNA from the nucleus, if you had integrated forms, theoretically, that integrated form could still support hepatitis D because it can produce surface proteins. And that's all you need for the hep D production and its replicate. So technically, even with hepatitis B cure, it actually has other implications from that front. But yeah, so the integrated forms are not technically infectious, and it's not necessarily directed insertion. So it's actually seems to be fairly random in terms of what's found. Of the liver cancers where it's been isolated from and checked from integration points. Again, every chromosome can be affected. Chromosome 19 seems to have more, whether or not they have more oncogenes that if it inserts in an oncogene, that one tends to create the cancers more often. But again, location of where it inserts, there doesn't seem to be any pattern. So it seems to be anywhere there's like a DNA nick or DNA thing, it's able to insert in there and do that. So, but it tends to cut in the same place because the linearization of the cccDNA occurs in the infecting stage. So when it's supposed to get converted from relaxed circular to closed circular, it has to go through a couple of steps, and if it misprimes, it creates a linear form. So it's part of its misprimed, misformed structure that is able to insert. So it has a similar insertion site or insertion direction, I guess, each time. So it cuts it at a very similar spot. Yeah.
Yeah. And any other conditions where you think there could be applicability of
for the integration piece with or for cleaving or for going after?
Well, the integration piece or going after the
integration. So going after the integration. So that one is going to be trickier. So the cccDNA by itself is like freestanding in its own little thing.
Yeah.
So it has a quadruplex there. If the quadruplex is in the other one, it may actually be a way of promoting the selectivity, because then we're not actually, if we're able to destroy the one that's free floating, that's great. But because it cuts it in a certain spot, we may be able to take advantage of that quadruplex that's hanging towards the end and say, you can only cut this one and not cut something that's integrated. So then we're not cutting our actual genome and our own genome. So it has from that front. Could there be ways of doing that? Maybe if we can anchor off that quadruplex and say, okay, now we're going to use an endonuclease that can cleave 12 nucleotides down, we could potentially cut it out of like integration. So there's ways around doing this in different ways from that. But now you have two free ends of DNA that could have implications in and of itself. So there's lots that have to go into this to be able to make sure that you're not creating more problems and gene editing and all of the challenges that come around that. So it's not completely straightforward, but I think it has potentials to kind of create new approaches and strategies to get people to kind of really think about what's in your field, what could be applied, and how can we bring that together, and how can we bring it to the virology field to be able to say, okay, yeah, actually that might actually work if we combine it with this. If that might actually work if we can figure out a way to overcome this other challenge. And so there's lots of potential.
Yeah, I
and I'll pull up the chat as well.
thanks. So firstly, I kind of think of myself as a relatively intelligent guy, and then I see this, and I'm like, whoa, this is way beyond me. Hep B's not got a huge animal reservoir, if I recall correctly. Can we eradicate it?
So it is a human virus. It is only in humans. And so we have passed it from human to human from, well, at least as far back as the 16th century, as far back as we know. There are mammalian models, but they're not great. There are animal models, but they're not all kind of great from that front. However, there's some that are better than others. We have a duck hepatitis B model, so an avian model, and that's actually what Dr. Lauren Terrell had used to develop the very first drug, lamivudine. So it was able to be used as a strategy for those nucleotide analogs. The one that the current more commonly used model now is woodchuck hepatitis. It also has a woodchuck hepatitis virus, but it is specific to the woodchuck. You can't infect humans with it. You can't take human hep B and infect a woodchuck, but it has a similar one. Yeah, I know exactly. And this makes it extra challenging because when we talk about drug development and drug studies, we talk about doing it in a mouse model. And so we're like, okay, well, let's infect a mouse. Well, no, mice can't be infected with this. However, this is where Lauren Terrell, in working with Dr. Kniedemann and Dr. Morris, they developed a hybrid mouse model. So it's an immunosuppressed mouse that you can give it human cells to create and to replace its liver. So now you've got a humanized liver inside that. So now you can infect that mouse. So there are some models, but again, it's a surrogate because now that mouse is immunocompromised. So it's not the full story. And this is some of the challenges of doing this type of work. But there are models there, and we have access to some of these things. But yeah, absolutely. Could it be eradicated? If we get rid of the cccDNA, sure. What I'm getting at from that front, I think going forward we can eradicate new infections, and I think that's the strategy that WHO is taking with that. Can we prevent new infections? Yes, we can. We have the tools to prevent it, so we need to use them, and we have to use them in the right way, in the right time to actually make it work. But yeah, so it is. I would argue eradicatable because it is. It needs to be a human virus, human only, so no animal reservoir, and but you also need good buy-in from countries or from governments and countries around the world, right? So to make it happen, but yeah, and then get rid of that other chronic form. Yeah.
There was an interesting question here. Lots of accolades, by the way, in terms of great presentation. Just in antenatal screen, only Hep B, I'm assuming surface antigen is done for screening. Triple screen is not done. Please explain what is the benefit of triple screening over just the Hep B surface antigen?
Right. Now that's a great point. So if a woman is hepatitis B surface antigen positive, then she very likely has the hepatitis B, and therefore the benefit of giving those vaccination and IVIG at that point is really important. So it is just a basic baseline screen. Doing your antibody screen tells you if you're immune or not, and doing the core antibody screen tells you if you've been previously exposed. That doesn't necessarily have implications for the mom giving delivery, but maybe down the road when she's about to start, anti-rheumatoid or biologics for doing all of this for arthritis that she's developed. Well, then that's when it comes into play, because knowing up front that she has that, then we already have it in mind. Okay, so now we need to make sure that we monitor, because even if it was in there, even if she's had a prior exposure, it could still reactivate. So yes, we do just have the straightforward the surface antigen on there. It would actually be fairly easy to incorporate that into this perinatal screening with that there. I mean, you already have the blood product and it just gets screened for the other ones. So that could be another area for advocacy in terms of just then people going forward, they know their status to be able to move forward and prevent and kind of go from there. So yep, thank you for that question. That is a really good point from there, just even in and of itself. But you're right, it is just surface antigen that gets done.
Any questions? Oh yeah, Pete.
There's a question about trying to achieve functional cure. Somebody's like reasonably young, healthy. Do you try to achieve functional cure in everybody, and how successful is that?
So that would be the goal, is getting functional cure. Although there doesn't appear to be anything that we're doing that's really pushing people to that. So it's about really just waiting sometimes and just. So the bottom line is we don't really have a strategy, or we don't really have a method yet that's actually pushing more people to functional cure.
But how successful is, like, what percentage?
Oh yeah, so people will spontaneously convert like about a % per year. And so there's, for some people, so in a clinic or in a population, like I'll we'll have conversations about, now we don't need to follow you anymore, once every few months I'll have a conversation, which is great. That means someone is no longer needing all of the.
But I mean, somebody who's had it since like they were born, like is there what are the chances that you could achieve functional cure?
Yeah, again, some of it comes down to, you know, some genotypes are more likely to actually create, to get to that stage. A is probably the best one for being able to reach that point. The chances, I mean, it's hard to really predict what it is. So when I see someone's blood work and they've had it since birth and now they're in front of me and they're 30, 40, 50, and they still haven't kind of had the antigens and the DNA levels trickling down. I can't give them that sort of confidence in saying, okay, well, you know what, in 1%, in 5 years you might have it. It's hard to give those numbers because we just don't understand why they're there, why they are what they are. And that 1% is just an all comers, all catch, and that's probably maybe it's swayed by some genotypes more than others. There's no specific profile, even that looking at this, there's a lot of work being done on biomarkers, you know, are there ones that are, you know, if we look at a broader term, a core-related antigen, if that is predictive of clearance and that way. But I can't give numbers, unfortunately, from that front. It's this catch-all, you know, 0.5 to 0.8% per year. People might spontaneously get to that point on their own. And so if there was a way, if we knew how it was working, if we knew how it was, you know, working better, we'd be able to give a bit more confidence in terms of our, you know, or even a strategy to be able, okay, we'll do this and it'll increase your chance by 2% or 5%. Or we just don't have some of that information yet. Yeah.
So one last question. I think it's a great practical question. When is it beneficial to give another HBV booster for patients with hepatitis B antibody that is low? And how beneficial are boosters when a full series has previously been completed?
Okay, great question. And this is one that comes up, and I think in the infectious disease world, we have a little bit of a different mindset, and I think in hepatology, they use the levels a bit more strictly. But I will say, so antibody levels that drop is a contraction of antibody levels. It's something that naturally happens because your body doesn't need to put in all the energy to make antibodies for a specific thing that it hasn't seen yet, but it still has the memory there. The B cells are still there. They're still doing what they need to do, unless you give them anti-B cell therapy. I know not some of you in the room probably do that, but I mean, the B cells are still there and able to make it. So just because the levels of the titers drop below doesn't mean you're non-immune, and therefore it is not necessarily, it's not recommended that you need to boost to get yourself above that level. And that's one of the challenges for some of this, because we like to react on some of those numbers, and actually it doesn't mean you're non-immune. Plus, if I were to then tell you, okay, you're an adult, even if you weren't immune, and exposure only leads to about 5% chance of it turning into a chronic infection, well, then you can already be reassured that, okay, you know what, I've got some antibodies, you know, my body is already primed to be able to see it, and the chances of that happening from that, and you would have to have a blood exposure that's fairly significant from well, okay, not from a blood exposure essentially is kind of the route of it doesn't necessarily have to be significant exposure, but if you're already immune,
Transplant.
yeah, a transplant. Well, yeah, okay, yeah, absolutely, that would count. But yeah, so if you have immunity, then the levels, the titers, we actually don't know exactly what titer equates to protection. You know, the correlates of protection, again, that keeps coming up in other viruses that we talked about in the past few years. But it is one of the challenges. But again, if you've been vaccinated, you are considered immune.
Thank you, Vanessa. I really enjoyed this presentation today because I think it also highlights for some of our young folks that the amount of energy and enthusiasm you have for your lab, and yet, you know, there's the clinical part that you related to is just, it's so nice to see. And, you know, so if people are interested, hopefully they will reach out to you in terms of having this clinician scientist career. And I know it's not easy, but it's wonderful to see your enthusiasm. So thank you so much. And before people leave, just an announcement. Next week, we have the editor of the CMAJ coming to do a combined rounds with the faculty. And there's also other presentations and so on. So if you get an opportunity, come in person. We're going to have some snacks, I hear, and coffee, but also there's other events that day. So thanks, everybody. Have a wonderful weekend.
Yeah, thanks, everybody.
You'll okay. Really good.
Thanks. No, I really like it.
So relatable.